बट वो अचानक दबसा गया, कुछ सही आंसर आ रहा था. What is DNA? That's very good. You need to track the DNA, right? And कल मैंने पढ़ाया इसीलिए मैं पूछ रहा हूं. And histone modification, somebody tell. DNA, I'm talking just in vitro. We'll come in vivo in few minutes. So you need DNA. You need histone modification or histone? What you need? Histone. So why these are not coming? So what is the definition of nucleosomes? Nucleosomes are basically you have a DNA and histones. So how many histones you need? Three or four or five or six or two? So histones are basic protein, right? So you need how many copies are there in a nucleosomes? 8. So 8 means how much? What are the names of those? So H3, H4, H2 and H2B. These proteins you need. Okay. And you also need with acetylation if you want to study H2K27 acetylation. So the question was in vitro. So how will you make the nucleosome? You need the DNA. Now it is clear, right? The question was you need the DNA, so you can make the DNA with the plasmid. You can use plasmid where 147 base pair of DNA is there. You can amplify it from the cells like any plasmid preparation you do, right? So you grow the plasmid which has a 147 base pair DNA cloned into that. So you have make a lot. In my program level, you make the DNA. Okay. So now it should be cloned in certain restriction site. So you cut the restriction site and then you will get 147 base pair of the DNA. You can purify and keep into the one test tube. Next question is you said, right, you need H3, H4, H2 and H2B. So how will you make it? I'm asking very easy questions. If you know cell biology, biochemistry, you can answer. So how will you make H3, H4, H2 and H2B? क्वेश्चन समझ में आ रहा है ना? मैंने बोला कि हमें H3 चाहिए बहुत सारा. PCR करके आपने क्लोन करोगे, ठीक है? क्लोन किया, मान लो आप in a plasmid that thing is there, then what will you do? So we can talk a lot about this, but these are the basic things and this is the last day. So answer should come up from you. How will you get a protein in vitro in a huge amount? Same cheese. You do the lysis. So what is this? This is called? So extraction of the protein. So how will you do extraction of the protein? Western blotting will detect the protein. Problem is you need to get H3, H4, H2 and H2B in a huge amount. Then only you will be able to study, right? So somebody is saying that they will purify from E. coli, right? This person is saying that purify from E. coli. Using recombinant DNA technology, that's what the term I was looking. Like in vitro, how will you make the protein, whether H3, H4, H2A? You do nowadays, you do recombinant DNA technology. You purify the protein. Okay? You make H3, H4, H2A. I am actually answering the questions for you. You should come with the thread. So how many of you think that recombinant H3, H4, H2A can be made and it is useful? My style, you know, right? Yesterday, the way I teach. Come up, raise hand. If you agree, raise hand. Only one person raised hand. And you answered that. So you also in the favor or not favor? प्रॉब्लम क्या है? प्रॉब्लम पे ध्यान दो ना. The problem is I need to study H3K27 acetylation or methylation in vitro. So first stage I cleared, right? You need a lot of H2A. The goal is to make nucleosomes. So you make DNA 147 base pair, keep it in the one test tube. I have kept it. Now I need 4 test tube, and somebody is saying that, sir, by using recombinant DNA technology, we can purify protein. All of us know. All of you have read, right? So you have a 4 tube which has H3, H4, H2A and H2B. So we are good now. So how many of you are agree that we are going in the right track? That suppose you have done gel and you have detected it, that's very good. You know that H3, H4, H2A has been purified. I'm not going in the steps, you guys just go and read. So that has been done. But question is, are we on the right path? So raise hand, how many of you think that I am on the right path?
For assembly, it needs some more enzymes.
Agree.
Will it happen in outside the cell of histone octamer and the DNA?
I say it happens for your sake of knowledge. We can make nucleosomes. I'm saying that. All this has been done, right? You can make nucleosomes, you can make dinucleosomes, you can make trinucleosomes, you can make all those using in vitro technique. Now, how many of you agree that we are on the right track? बहुत सारे लोगों को convince कर पाए. Numbers are good now. But how many of you think that I am not on the right track? Why? Or you are confused, you don't want to. You have to pick side, right? कुछ आवाज नहीं आ रही है. क्यों disagree है? कुछ भी तो सोच रहे होगे ना? Let me know that your thought process. Don't get shy. I'm telling you, I can teach you everything. There is no point of going and learn about therapy and different hypotheses if you do not design a simple experiment in epigenetics. That's what I'm trying to teach. Yes. हाँ.
पोस्ट ट्रांसक्रिप्शन मॉडिफिकेशन नहीं होता है, फोल्डिंग हो रही है ठीक से कि नहीं हो रही है, और हम ये श्योर भी हो सकते हैं कि वो प्रोटीन जो बना रहे हैं वो फंक्शनल है कि नहीं, तो हम ये कैसे मेक श्योर कर सकते हैं?
ठीक है, fair enough. You can answer in Hindi also. There is no need to answer in English. I have to speak in English because that's I think. But if you know Hindi, you can also tell in Hindi. Not a problem. Yes, I totally agree with your confusion, and it is a valid question. Then now other people have to defend, right? All of people raise the hand. They have to defend that. Why do you think that you agree? Anyone? Because I said, then you agree, right? I'm teaching wrong thing, then you will agree? So there is a reason you raise hand, right? Anyone can answer why you raise hand? You thought that that is the good way.
We can extract different types of protein in different from different cells. So that's why I agreed. Like we can lyse the cell and then we can take them.
But he is telling that it will lack PTMs. Then now you need to change your strategy.
We can do the induction for the.
You have done, right? I already we passed. Well, you have a S3, H4, H2A. Think this 4 chair as a H2A. You have a lot of H3, you have a lot of H4. Just you have make using recombinant DNA technology. You overexpress the protein, you purify it, and you have kept. You identified, characterized everything. But he is saying that, sir, वो तो होगा नहीं, क्योंकि we lack the modification. Then what will you do then?
We will put the external something to modification of that.
Okay.
Then chemicals we can use like something we have added to that.
You can add methyl transferases and you can make the methyl group. Okay. Then, but I say that I will go with the recombinant only. So to tackle your problem, what will be your approach then? How will you purify H3, H4, H2, H2B? You need modification, then how will you purify that? Okay. Okay. So now how many, I got it, you need to purify from cell line or from any system you directly purify. That is also doable. Now I leave you I following him. So how many of you agree with him? You also agree, change the answer. So how many of agree with him? What is your name? Shailendra. How many of you agree with Shailendra? What is your name? Shivani. 2 people are leading now. How many of you agree with Shailendra? Raise hand. I will not able to understand. इसमें कोई एग्जाम तो नहीं चल रहा है ना, आप ही का समय जाएगा. If you do not come to the decision, not taking decision is the worst thing you can do in the life. I'm telling you, we all the time wonder we are on the right path or wrong path. Just take a decision. तो 2 मिनट 2 सेकंड लगेगा, रेज कर दो शैलेंद्र के अगेंस्ट में या उनके फॉर्म में. हाउ मेनी ऑफ यू सपोर्ट शैलेंद्र?
Sir, if we are doing modification in yeast, it will be not our desired modification. So we have to, if it is undesired, we have to remove it, and again we have to go for writers and erasers in the new in vitro technique.
So you don't like Shailendra then, right? So you don't.
No, I have just confusion. If the modification is not our desirable, so what we can do?
So what is your name? Huh?
Because
Vikas. So how many of you agree with Vikas? One person. Why? Other people disagreeing him. It's a valid point. Isn't it? When you are purifying from the cell, histone H3, H2, and H2B, it could have a number of modification. And I told you specifically the problem is I want to study H3K27 acetylation or H3K27 trimethylation. Then he is raising the valid point. Ki, sir, when you purify from the system in vivo, you have a lot of problem. Because in a cell, if you are doing Western blot, you could be acetylation, methylation, at whatever acid you want. You learn to write 10 days last we are studying. So, Vikas has now raised the point. Now what do you think? Now how many you agree with the Vikas? Is number changed? Just take the decision, it's a fun. I'm not judging you again, If you not raise hand, it will be it will not be a fun class. You want to be just go to the lectures? No. So please take decision, because we as a leadership, you will be future leaders. You need to learn to take decisions. Not taking decision is the worst thing. Not agreeing is the worst thing. Confusion is the worst thing. Disagreeing is a good thing. Agreeing is a good thing. Not being confusion is the worst thing you can do in your life. So how many you agree with Vikas? Okay, at least 1 or 2, 3, 4 people came up. That's very good. So he has raised valid point, and she has also raised valid point. So, and he has also raised the valid point. So it become context. So when you are studying in vitro, then you don't want other modifications to come into the picture. Why? Because then it will complicate the situation. If you are focusing just for the role of H3K27 acetylation, then it will be a difficult thing. If you purify from the cell line. So the in vitro experiment, you need to make the histone in the recombinant form. And then there are different ways through which you can put acetylation. Now people, you can put the acetyl transferases, you can specifically take the enzyme which do H3K27 acetylation. There are enzyme available, you can buy that. You make the nucleosomes, you put that, you can do that. So how will you make the nucleosome? If you do not want any modification, so you have a 4-tube H3, H2, H2 and B, purify it in a denaturing conditions and put the DNA, and through the salt gradient, you can make the nucleosomes and you can do gel filtration, all those study. Okay. So this is the one way if you are just focusing for K20. So for the in vivo study, you need different kind of setting. You can use antibody against H3K27 trimethylation, H3K, where you have all the component will be there. So it depends on what kind of questions you are answering. Okay, so in vivo, if you purify histones, then people go for, if you directly want to probe, people go for the recombinant DNA technology. Okay, then you have a very defined system. But if you purify from the system, it is still confused because there could be problems. You have other modification also. So the phenotype you will observe. So the people who will make the nucleosome with the cylinder approach will get one phenotype, and the people who will make nucleosomes with your name Shivangi approach, you will have another phenotype. Okay, because there will be heterogeneity. Now question is clear or not clear? Okay, any doubt? So we took a lot of time to make you understand that. So in vitro, you can address different question. In vivo, you can address the different. That point I want to make. So actually, because of these modifications in the cell, you get, although the genomic complement is same, you will get the different DNA. So this is basically telling you, you have read all about this. Like there are different type of epigenetic mechanisms, and since this class is putting toward to understand that therapy, so you have your therapy could arise from either non-coding RNA, you can arise through chromatin remodeling, you can come with the RNA modifications, you can of course come through the DNA methylation. So I taught you a lot about DNA methylation yesterday. So and then the therapy can come to this picture. So when you are thinking about the therapy, you have to figure it out that your epigenetic drug could target any of this pathway. So clear or not clear? And this is just showing you the cartoon where you have a euchromatin also, which is highly transcribed. You have heterochromatin also, and then you can have a nucleosomes which can evict you can slide all those things you can do, and you have also enhancer and promoter. Okay, that can also play the role. So to target this pathway, you need to really go down and figure it out that which enzymes and which proteins are actually important, okay, for your case of study. So these are very new. Now people are also coming to the RNA methylation stuff, which is also important to the epigenetic mechanism. So this is taken from this review, which has very extensive information if you want to learn about this. So now in your system, you know that what pathway, what thing will going to affect. So this one is also a complicated slide, but the point I want to make that just focus on one thing and you need to figure it out which pathway, and then you can go for the drug design. Now most of the time I will take the example for cancer, since we study cancer epigenetics a lot, but you can apply the same concept even if you are studying other disease type, if not infectious biology, or you can apply the similar concept in there also. So the talk focus will be on the drug which is important for the cancer treatment. Okay, so these are the different pathway, because I took cancer because in this situation almost all epigenetic modification has been shown that they have been involved. Involved means chromatin modifiers are either mutated or overexpressed or downregulated, or they are in coming with the infusion if with some transcription factor. So those who are not in cancer biology, the point I am trying to convince you that the things we talked about DNA methylation, so there will be enzymes to do the methylation, so that might go up and down, that might go mutated. If you are trying to understand that what is the chromatin remodeling, then that protein which does the remodeling activity could involve. So there are tons of things which you need to figure it out, and this is the outside showing that it could be global DNA hypomethylation, and I teach what is global and what is local, right? Because you can just take the cancer cell type normal counterpart, you can measure the DNA methylation and you can say that there is overall change or not. And then if you want to do local, then I also explain you that you can purify locus specific and then measure the DNA methylation. Okay, so when I'm saying global means there is a global change is happening. Similarly, many enzymes could be affect the oncogene or tumor suppressor function. Also, there could be mutation in RNA that could also affect the oncogene or tumor suppressor function. So these are basically all these mutations arise due to the after the formation of zygote. So they are basically the somatic mutations, and there are many examples in the literature. So what I teach that chromatin modifier, whether it's a acetyltransferase or deacetylase, whether it's the S3K27 methyltransferase, any methyltransferase or demethylase, they could be involved in the activity of tumor suppressor or oncogene, or they could target the genes which is responsible for the oncopotential or cancer phenotype. So like cancer phenotype is what? Proliferation, increase in proliferation, then increase in invasive property. So this histone modification or chromatin modifying enzyme are actually acting on some of the proliferative enzymes or some of the enzyme which regulate the cell cycle, and then you can control their expression, and ultimately we call the oncogenic potential. So clear, right? So now you know that you have a gene involved in cancer. So if you want to say that epigenetic mechanism are there or not, what will be your approach? Suppose I am saying that H3K27 methylation. The name of the enzyme I will tell you, EGS2. That is involved in changing the cancer phenotype. How will you design the experiment? What will you do? Very good. And then if you find if there is a hypermethylation, then you can cross correlate. You can, if you do not have a sequencing information, what will you do? My lab is not have lot of money. Then I said that no, Vikas, right? Salender, sorry. I forget the names. So sorry. So Salender, I said that I don't want to do sequencing. Figure it out some other way. How will you do that? So you understood the problem. So the problem is, suppose this bench is the gene. Okay. Now we know that the promoter of this has been decorated with H3K27 trimethylation. He is saying that by chip sequencing, and this cell line. How will you prove? He said he will do the chip seq. I said that no, I don't have money. Then you will fight with the PI and leave the lab. That's the worst thing you can do in your life. Because time is important. You work for 2 years with him and he is suggesting that we do not have the reagent. Then you will just stop. So this is the biggest problem in Indian science, because you have to come up with the smart answers and smart technique where you can use less money but answer the questions. And believe me, there are answers available. So I am saying that there is no need to do sequencing because I want to get the targeted approach. So how will you do it?
Can do means check the hypermethylation is occurring or not by pulling down the DNA methylated part, and by quantifying the protein, by quantifying the protein, it is increased. We can say that it is hypermethylated. If we bring the hypermethylated protein more, it will be.
So you are saying, what I am understanding from your answer, you are saying that you use antibody against H3K27 trimethylation. You pull down.
If more pull down occurs, it means more hypermethylated. Means the site is more hypermethylated.
But you are doing in the whole context, and it could possible that it is just working on one gene. You can do that approach if it is very high H2K27 trimethylation. Then you can, of course, purify and you can know what are the proteins are there. You can use that approach. But if you see, you did the same thing with the cancer cell line and also the normal, and you see that there is nothing going on. There is no protein. What will you do then? How will it going to help you?
How much are acting on the specific gene? Like in ELISA, similar to that, we could see how much the.
Okay, you can check for the functionality, right? That is the answer. Yes, you can check for the functionality. but I want to probe it at the chromatin level. How will you do that? Just we don't want to do sequencing. You can use any. How will you do chip? How many of you understand the chip assay? Raise the hand. How many of you know about transcription, how to study transcription factor? So in a chip experiment, what you do? You have a chromatin, you need the antibody against that, right? Whether the modification or against the protein, right? Then you do the sonication. So you clip the DNA. Then from that complex, you purify the DNA. And after purification of DNA, what you do?
Sequence the DNA around the nucleus.
I said that sequencing will not, I don't have money to do the sequencing. I want quick amplification. How? Now I am totally confused. Somebody is saying PCR, somebody is saying RT-PCR. Again, I have to do this. How many of you think PCR is a good technique? And how many of you think that RT-PCR is a good technique? You are raising hand in both. Huh? Though both RT-PCR and PCR. How many of you think that RT-PCR, qPCR will be a good technique? These people are not raising hand. Why? So I am trying to give you the thought-provoking thing. So see, today's lecture, we talk about therapy. The drugs are available, but how will you going to design the drug? This concept you have to understand, right? I'm just giving about example of STK27 trimethylation. कल को आपको ड्रग डिजाइन करना है तो ये सब पढ़ के कोई फायदा नहीं है क्योंकि वो तो ऑलरेडी डिजाइन हो गया है. हमें नीड टू design our assay system. So to design the assay system, you can play it out. So now you know that somebody is saying that you can do chip sequence, and then you know that how many blockers this particular STK27 trimethylation is there or not. But I said that you need to do with the not with the sequencing, just so somebody said that PCR, somebody say qPCR. So there I am stuck now. Which one is the best? All of you are facing an interview and the guy is asking which one is the best. What will you do then? So in PCR क्या करते हैं और qPCR में क्या करते हैं? सोचो तो पता चलेगा. Okay. So the whole purpose was whether that K27 trimethylation is there or not. So if you do the PCR, then it's not going to solve you. You need to do the qPCR. So that is what we do in the chip. Any locus, if you want to see the enrichment of a protein, you will do the qPCR. Okay, I'm not going in detail because I assume that you have learned this during 8-9 days. So that's why I'm trying to touch the hint and then move forward. Okay, so you can do the qPCR because you know the DNA sequence. So as I said that there is no need to do the chip-seq. So now you will agree with the PI. No, I can do sir experiment. I will not leave your lab. Because I know that you can use, always think about the alternative technique. Even the fancy things are available. How alternatively you can prove. He is also right. You can do that thing also. People, it's called it like a pull down the whole locus, do the mass spec and see the differences. But it could not find differences. Then you can use this technique also. Clear? Now, if you are suspecting that EGH2 is the enzyme involved for this, now you can design the inhibitor and test, see the experiment you can do now, because you can use the inhibitor of the EGH2 with and without, and then measure K27 trimethylation by chip. You can measure the cancer phenotype using any proliferation assay. So it's clear, right? So you can take the domain which is involved in to do the catalytic activity. It could be catalytic independent activity could also be there. So in this way you can do the drug design. Now I have put again complicated slide that, yes.
If we design one inhibitor, it will affect all the readers and writers. No?
So if it is specific to that protein, I agree it could affect other also, but that is the whole point. You need to design very specific inhibitors. So that's why if you want to check all the effect, then you will do the ChIP-seq and other to see the pan. If you want to see first at a gene level what is happening, then you can test that and modify accordingly. Okay, got it? But yeah, that you have to test it, whether how specific it or not. So that is the problem of chemical structure, biochemistry, all those problems. Okay, but you can do that. Now, so far I have teach you that, telling you from yesterday that heterochromatin is very, very important. Euchromatin is very, very important. So can you experimentally prove that heterochromatin is important for